So this is covering exercise 40, the anatomy of the urinary system. So the main reason why we're covering the urinary system and why we have this system is that it is going to be primarily concerned with the removal of waste. So as we have all these biological processes that happen in our body, we will create a lot of waste. So the urinary system will be honestly, it's the trash dump system to be able to get rid of all of that waste. Specifically, what we're really talking about is liquid or almost liquid waste. The digestive system will be also getting rid of more solid waste. So this is going to be liquid, and the most of the waste that we need to get rid of will be nitrogen. Some sort of nitrogenous waste is going to be the main thing that we need to get out of our body. While these are the main functions, there's a couple other things that your body uses with the urinary system to kind of keep homeostasis happening. And that is, it was also responsible for fluid balance, which blends into this removal of liquid waste. So if we have a lot of fluid in our system and we need to kind of take away some, the urinary system will take care of that. If we need to keep some liquid because we don't have enough liquid, the urinary system will also do those. So those also go together. And that's a way that our urinary system can maintain homeostasis. Another way it will actually do it is that your kidneys are responsible for maintaining the acid-base concentration in your body. So we've talked a lot throughout AMP 1 and 2 about acid and bases and certain things that need to have, like the digestive system needs to have an acidic environment, or some other things need to have a basic environment. So overall, your urinary system really does set the stage for maintaining the acid-base concentration in the body. So when we're talking about the urinary system, one of the main structures that we'll talk about, or the main structure, is going to be the kidney. And we'll look at this both at the gross scale and then at the microscopic scale. And with the kidney, this guy is really cool. He does a couple major things. The first one is he's going to be a blood filter. So your kidney will actually act as a filter for all of your blood. But then it's also a blood processor. So not only will it filter out all of the blood, but it will also process it and clean it up and do all the things it needs to do, put things in order to maintain its integrity. And then it will also do, once it's got these two things underway, our kidney is responsible for manufacturing your urine, which is that liquid waste that we need to get out of our body. So the kidney is going to be our main structure, but let's look at just the overall urinary system structures. So the main structures we're going to have, if we have our structures that we're going to have. Here's our little person. When we're talking about the urinary system structures, we're really going to be talking about anything that's on the lower half of the body. So the first one we're going to have is you are going to have your renal arteries. You're also going to have your renal veins. Arteries are carrying blood away from the heart, so these are going to be carrying blood toward the kidneys. And your renal veins will be carrying things away from your kidneys. So this is how we filter the blood. Renal arteries will bring the blood toward the kidneys. The renal veins, after it's processed, will take it away. There's also going to be 2 ureters. There is going to be 1 urinary bladder. There's going to be 1 urethra. And there's going to be a structure called the external urethra orifice. This is how the urine will actually leave the body, the external opening of the urethra. This will be different based on male versus female, but they generally have a structure that will be, not generally, they will have a structure that's the external urethra orifice, but it will be structurally different based on male and female. So we're going to start with these guys on our lovely little picture. So one other thing I totally forgot is you have 2 kidneys. I covered them above. I forgot to include them. 2 kidneys. So the way this is worked in the body is on either side of your spine, you will have kidney 1 and kidney 2. And they are kind of shaped like kidney beans. There will be 2 kidneys. Leaving your kidney, you will have a structure that is going to be your ureter. So in this one, it's going to be red. You have 2 ureters. Those are these guys. Those ureters will dump into one urinary bladder. That one urinary bladder will then have the urethra that will then lead to the external orifice and dump out of the body. So this is kind of how it looks if we were to blow this up and take it off of our little person. These structures look, here is our midline of the body. You're going to have your 2 kidneys. We're going to talk about these next in depth. You're going to have your ureter from each. These are off the midline, you'll notice, and then they will dump into your bladder, which is on the midline, and then that bladder will release from the body through your urethra. So we will have one on each side that will then dump into the general bladder. So now we're going to blow up the kidney and really look on the inside of this structure. We're going to look at both the microscopic and the gross, specifically of the kidney, which is the primary urinary structure. So these are going to be the exact same whether or not it's on the right or the left, and they do have the general structure that looks like this. So we're just going to do one, but they are the exact same. There's no differences as of right now that we know the right or the left. So when you're looking at the kidney, there's actually going to be 2 main regions. You are going to have the renal cortex and the renal medulla. 2 main regions. So when we're looking at the kidney like this, the renal cortex, inside you kind of have this structure. This part right here is going to be the renal cortex, whereas this part on the inside is going to be the renal medulla. If you think about the brain, where we had the cortex of the brain, it was the external, and then the medulla was going to be more of an internal structure. So if that kind of helps, it's the same flow with the cortex is on the outside, the medulla is on the inside. So inside the medulla, what you're going to have is a couple other structures. You're going to have these things called renal medulla pyramids, and they will fit into these little structures, kind of like this. Pyramids, because they kind of have a ish triangle because they have to fit in this little spot. So these are going to be your renal medulla pyramids. Then what you're also going to have is inside this structure, inside these renal medulla pyramids, we're going to take a cross section and look at this and look at what happens when you are looking at the section between the medulla and the cortex. We're not quite there yet, but we will be getting there. The other thing I want to talk about when we're looking at the kidney is the medulla, the cortex, the renal medulla pyramids is going to be how we get the blood in and how we get the blood out, because that's the entire premise of our kidney, is to be able to filter the blood. So we have to get the blood in and we have to get the blood out. Coming into your kidney, you are going to have a structure that will bring blood in. So this will be your renal artery. This will bring the blood into the kidney. Then you will have a structure that then takes the blood away from the kidney. And this will be that renal vein. So basically blood will come in the kidney, the renal artery. It will get filtered in the kidney, and then the waste products will, or the blood will actually travel out through the renal vein. You also then are going to have a structure that is going to be leaving the kidney with all of the waste products, and that's this guy. So the kidney up here, when we were leaving it, we were leaving through this structure right here, which was our ureter. So coming out of our kidneys, there are 3 connection points: blood coming in, the artery, blood going out, the vein, and then the waste products being removed, and that is going to be your ureter. So it generally looks like this structure on both sides. Your kidney will actually filter 1/4 of your body's blood. It will filter about 1/4 of your body's blood that will flow through the kidney in under a minute. So in 1 minute, 1/4 of your body's blood will flow through this artery and out this vein and get filtered by the kidney. So that means that's a ton of work that your kidneys are going to need to be doing to do essentially in every 4 minutes. Your entire blood supply will get filtered at some point. Okay. Next, we're going to take what we said was this lovely little section right here, and we're going to blow it up. And that is the connection between the medulla and the cortex. So we're going to draw it right here. So when we have this little connection point right here, we're going to say this top. This is going to be a really big drawing, so that's what I'm trying to figure it out. We're going to have our cortex, which is the external layer, and the medulla, which is more of that internal layer. So the way we're going to start this process is the way we started the kidney. Blood is going to come in, and then we're going to have blood flow out. So what happens here is blood will flow in through a structure called the afferent arteriole. So blood will flow into the body through this tube. This tube will dump into a structure that looks like all of these tiny little capillaries. And then it will have a tube that goes out, and that will be the efferent arteriole. So essentially what this is your afferent arteriole will bring blood in. This lovely little conglomeration of structures right here is going to be your capillaries. These capillaries have a name. This is going to be called your glomerulus. Your glomerulus is just capillaries. You have all of your blood in here going through, and then it will then, whatever, as it travels out of the kidney, it will go into the efferent arteriole. This glomerulus is going to be a collection of capillaries. What is happening is right next to this glomerulus, you will have a structure that looks like this. And this will be called the Bowman's capsule. The Bowman's capsule, and it will have tiny little holes in it. So it will have these lovely, they're supposed to be little holes. These holes are going to be called fenestrations. Essentially, it's going to act as a sieve. So if we have blood flowing into our glomerulus, going through our capillaries, as they pass through this glomerulus, as they pass through this Bowman's capsule, it's going to act as a sieve, and it's going to push things from the blood out into the Bowman's capsule, and the Bowman's capsule is going to collect it. So the easiest way I really like to talk about this is if you ever played with Play-Doh when you were a kid, and if you took some sort of structure that had a bunch of holes in it, let's say you're going to do it with a a sieve, and you push that down onto the Play-Doh, and you get all of the little spaghetti noodles that pop up. That's essentially what's happening here, is you're putting this structure with a bunch of tiny little fenestrations or little holes on next to this liquid, and you're going to have some of the liquid inside our blood come out and go through this next process, whereas you're going to have the main part of your blood left over, or the main chunk of Play-Doh will still be on the other side, and then that will then get escorted out of the kidney through the efferent arteriole. So one thing to keep about this is when we're looking at this structure with the glomerulus and the Bowman capsule, we are not filtering out cells. Your red blood cells should not be filtered out. They should stay within the blood. Your white blood cells should not be filtered out. They should stay within the blood. What we're filtering out is the plasma. So it's the liquid that is going to be housed as part of that whole blood that is going to get pulled out. That is what we're filtering, not the blood cells themselves, either red or white. We are filtering out the plasma that makes up the rest of whole blood. So the plasma is carrying the nitrogen, the nutrients, the gases, some of the wastes. So that's what we're needing to filter, not the actual cells themselves. So with this glomerulus and this Bowman's capsule, this structure together, these two, Bowman's capsule and glomerulus, are going to be called the renal corpuscle. And if you look at it underneath the microscope, it really does look like a ball of yarn, or a collection of blood vessels. So this is your renal corpuscle. It's the connection between the capillaries of your glomerulus and then the case around it, which is the Bowman's capsule that is filtering everything out. Okay, so now we have the blood going through our glomerulus. We have filtered out and we have collected. Now that we have collected all of the stuff, it is going to dump into a tube that looks like this. That tube will then look like this. And then that tube will look like this. So this is the general structure of your nephron. I'll name that in a second. So as we have all of this stuff coming out of your renal corpuscle, it is getting collected, and what it will actually do is it is going through all of these tubes. It will go down this loop, up this loop, and then it will go through all of these tubes until it is going this way. So it is a one-directional system. So coming off of the renal corpuscle, this section right here is going to be called the PCT. The PCT stands for proximal convoluted tubule. And if you look at it, it is convoluted and it's a bunch of tubes. Proximal is because it's closest to the nephron. It's closest to the glomerulus. This one right here is going to be DCT, which stands for distal convoluted tubule. DCT. And again, it's distal to the glomerulus, so that's why it's got the DCT name. So as the liquid in our blood, our plasma, is filtered out, it will go into our PCT first through the process, and then it will go down this. This is going to be called the loop of Henle. I'm sure in lecture you're going to talk a lot about the loop of Henle, but we're just going to name it right now. So this is the direction that the filtrate is going. You'll notice that the loop of Henle is going to be with it's a one direction. So this part will be the descending loop of Henle because it's going down. This part will be the ascending loop of Henle because it's going up. And then we have all of this DCT, and he will dump into a general tube that is going to be called the collecting duct. Actually, let's do that as green. This is the collecting duct. And it is just what it says. It is collecting things. So it will get information from this nephron. It is also getting information from another nephron down the way. So he is going to be essentially the public sewer system that is taking away all of the waste. So this is what we call this structure right here is called the nephron. And the nephron is going to be the part of the kidney that will actually filter blood. So this is what it looks like. Or it will actually filter all of the plasma. Your nephron is the main structure that will take care of that. And this is how it works. So blood will come in through the afferent arteriole, go through all the capillaries that are called the glomerulus. Whatever liquid gets pulled out, filtered out, will be filtered out through the Bowman's capsule. The rest of the blood cells, and any other solid structures will leave the nephron through the efferent arteriole. With all of the liquid that we have pulled out of the kidney, if all the liquid we've pulled out of the blood will get filtered by the renal corpuscle, and it will dump into the proximal convoluted tubule. It will go through that process of tubes. It will travel down the descending loop of Henle. It will then travel up the ascending loop of Henle. Then it will go through the DCT, or the distal convoluted tubule, and then it will go and dump into the collecting duct. This collecting duct right here, at this point, this will be called urine. So during this process, it's not called urine, whatever this liquid is, it's called filtrate. The liquid that has been filtered out is called filtrate. Once we get to the collecting duct, that liquid is now called urine. Okay, so one other thing with these nephrons are two types of nephrons. You are going to have a nephron that is called a cortical nephron, and then you will have a juxtamedullary nephron. So a cortical nephron, if you think about the name cortex, a cortical nephron will be mainly in the cortex. So that means when we drew this thing originally, this was the demarcation between the cortex and the medulla. I kind of wrote over it, but let's do this. This was the demarcation between the cortex and the medulla. What we're saying with a cortical nephron, the loop of Henle only juts down a little bit into the medulla, but the majority of the nephron is in the cortex. But then you're also going to have one called juxtamedullary nephron. Juxtamedullary. And this one will be further into, and it will penetrate the medulla. So a large chunk of the loop of Henle will fall into the medulla. Keep in mind the top part, the PCT, the DCT, and the glomerulus will be in the cortex, but it's only the loop of Henle that will either come a little bit into the medulla, or it will come pretty deep into the medulla, and this would be this juxtamedullary nephron with a deep loop of Henle. I'm going to get rid of that. Okay, so one other thing about this entire process that is really, really cool is this whole process was responsible for filtering blood, and it was also responsible for producing urine. We have not talked about either two of those processes yet. So the first one we're going to talk about is the juxtamedullary apparatus. Do not confuse that with the juxtamedullary nephron. So this is the juxtamedullary, erase that. The juxtamedullary nephron. This is the juxtaglomerular apparatus. Juxtaglomerular apparatus. Juxtaglomerular apparatus. This is responsible for regulating the blood filtration. So this will actually turn it on and off. There's a process to control the blood filtration. And it's also responsible for controlling the systemic blood pressure. So your heart will be involved in blood pressure as well, but your juxtaglomerular apparatus is responsible for your systemic blood pressure. And there are two structures to this guy. The first structure is going to be called your JG cells. Depending on the edition of your book, they could also be called granular cells. I have seen both used. So JG cells or granular cells. These are going to be blood pressure sensors. Blood pressure sensors. And they are going to be found in the afferent arteriole. So a blood pressure sensor in the afferent arteriole. So that means way up here in this guy, all the way here. This is going to be our JG cells or our macula densa cells right here. These are a blood pressure sensor. So our juxtaglomerular apparatus has JG cells that are a blood pressure sensor in the walls of our afferent arteriole. So that means as blood is coming in, your JG cells right here are attesting to the pressure of the blood. If too much pressure is coming in, it will trigger your system to decrease that pressure. If not enough pressure is coming in, then it will trigger your system to increase the pressure, because what it cares about is when blood is traveling into your afferent arteriole and it's going through this glomerulus process, the pressure of that blood moving through those capillaries needs to be correct. If there is too much blood, too much pressure forcing that blood to travel through your glomerulus, essentially too much pressure of the blood means you are not going to get enough liquid filtered out. Whereas if you have a low pressure coming through your afferent arteriole, your glomerulus is going to have too much liquid for it to be able to process out. So the systemic pressure of the blood flowing through your arteriole and flowing into your glomerulus is highly, highly important to be able to filter out what it needs to. So your juxtaglomerular apparatus is going to be responsible for doing that through the JG cells that are found in your afferent arteriole. A second structure for the juxtaglomerular apparatus are going to be called the macula densa cells. Your macula densa cells. And these are going to be a chemoreceptor. So they're going to measure chemical concentration and chemical level. Chemoreceptor, and these are going to be found in the distal convoluted tubule. So it's a chemoreceptor that is found in the distal convoluted tubule. So if this guy right here is labeled as our JG cells, then our macula densa cells in our DCT are going to be labeled like this. So coming all the way back up here to our nephron, we're going to have our macula densa cells. And our macula densa cells are a chemoreceptor. The main chemical that they are picking up is going to be the guy who's responsible for pH. So he is looking at the concentration of our hydrogen and the concentration of our OH, mainly this guy. So your macula densa cells is picking up on the concentration of your hydrogen ions in your distal convoluted tubule. Mainly what that is doing is, if you have gone through this entire process of filtering out blood, having the filtrate, which is just the liquid from the blood, travel through the PCT, loop of Henle, and DCT, what's happening is it's taking things out of the liquid and putting things into the liquid. We're taking things out that we want to keep, and we're putting things in that we don't want to keep. By the time you get all the way down to this, you've gone through here, you've gone down here, you've gone up. By the time you get to the DCT, you are getting ready to dump into the collecting duct. That means that whatever the concentration of this filtrate is, it needs to be the right concentration. So by the time it gets to the DCT, if there is too much hydrogen ions in it and our body needs some more, we will take some of those hydrogen ions out. If we get to the DCT and we have too many hydrogen ions in our system and we need to lose some more, it will pull back and put some more into the filtrate. So this macula densa cell in the DCT is a chemoreceptor who's mainly picking up on the concentration of hydrogen ions. So this is the nephron as a whole. We've got two different types of nephrons, the cortical and the juxtamedullary. We've got this thing called the juxtaglomerular apparatus, which does blood pressure and blood filtration. But one thing we haven't talked about in this entire process is how you actually form urine. And we're going to go through this, and this is where we are going to end for this exercise. So how do you actually form urine? It's a 3-step process. So first you will go through a process of filtration. You're going to filter out from the blood. Then you're going to go through a process of reabsorption. And then you're going to go through a process of tubular secretion. So let's break these down. And then at the end of this, you are going to have urine. So the first step is going to be filtration. So filtration is going to happen at the glomerulus. So we'll go up there and look at that in a second. It is a passive process. So what you are doing with filtration is you are simply going to take the part of the blood from the capillaries and you are going to filter it into the proximal convoluted tubule. So at this point right here, the blood is going to go from the glomerular capsule and it will get filtered into the proximal convoluted tubule. And at this point, it is now called filtrate. So we have filtered. So all the way back up here, what we're saying is the glomerulus, this part right here on this nephron, this part right here, where we had afferent arteriole coming into our glomerulus and out, this is going to be the process of filtration. We are filtering out things, filtering out the liquid from our blood through our glomerular capsule into our proximal convoluted tubule. The next part is going to be reabsorption. And with reabsorption, it's going to be tricky to think about. So reabsorption will occur at the PCT. And what is happening here is your filtrate moves through the PCT. So your filtrate will move through the PCT and it will return whatever things it needs to return to the peritubular capillaries. So that's something I haven't actually talked about yet. So your filtrate will move through the PCT and will turn items to the peritubular capillaries. Okay, so first of all, what I want to talk about is, here is blood coming in, blood going out, our glomerulus, Bowman's capsule. I'm just going to redraw it. And then we've got our PCT, loop of Henle, DCT, collecting duct. So that's kind of our general, here is our, we're just going to get rid of that. We don't need that right now. That is our PCT is right here, and our DCT is right here. So this is just a general quick look at this process. We talked about this part right here with blood coming in and blood going out. This part was filtration. What we're now saying is reabsorption is going to be the PCT. So we're going to be talking about this part is reabsorption. And what I mean when filtrate moves through the PCT and will return items to the peritubular capillaries is we are saying that as things are filtered out over here, and it comes through here, and then it goes through this process. Let's say that what we filtered out right here was a hydrogen ion, and then it got put through here, it's a hydrogen ion, and then now we're here in our PCT and it's a hydrogen ion. But wait, our body wants to keep a hydrogen ion. This is where it gets tricky and where it gets weird. So coming off of your efferent arteriole, coming off right here is going to be a capillary. That capillary is going to be called the peritubular capillary. And this capillary will essentially do this. So that means your peritubular capillaries, you will have a blood supply around the entire nephron. So when we're saying that the filtrate moves through the PCT, so the filtrate moves through the PCT here, and it will return items to the peritubular capillaries. So we have this hydrogen ion in our PCT, but wait, we don't need it. You can actually put it back into your peritubular capillaries. So you are essentially first filtering things out of your capillaries, and then you may need to reabsorb them back into your capillaries. So reabsorption is from the tubes or from the PCT. So your for reabsorption is going to be from the PCT to the capillaries. You were going back into the blood supply, whereas filtration was coming from capillaries to the nephron. This one we're going backwards. We're going from the tubes back to the blood supply. So reabsorption is you are reabsorbing items back into the blood. So there's a couple trends that will follow with what things want to get absorbed. So with the process of reabsorption, there's a couple things. You will have water occur. Water will reabsorb through the process of osmosis. And then you will have some things that are going to be almost entirely reabsorbed. Depending on your body system, these things will be reabsorbed completely. Water, glucose, and amino acids. So these three will get filtered out through the filtration process, but then when they get to the reabsorption process, your body takes them back and reabsorbs water, glucose, and amino acids. There's some things that are selectively reabsorbed. selective reabsorption. Depending on if your body needs them, you will reabsorb them. And a good one of this is ions. So if you've been working out and your body needs to maintain your ions and not get rid of it, you will reabsorb your ions. If you have done nothing but sit on the couch and eat a bag of chips, you've got a lot of ions in your system, your body doesn't need to hold on to them, so they will probably keep them out. So you will have some selective reabsorption. And then there's some things that you will always want to treat as waste and get rid of. And that will be urea, which is a nitrogen base, uric acid, which is also a nitrogen base, creatinine, which is a byproduct of cellular respiration and being a oxygen consumption, and then any kind of drug metabolites. If you're introducing a chemical to your system through some sort of chemical pharmaceutical drug, there's always going to be parts that you will need to get rid of that your body doesn't need. So those are called drug metabolites. So these are the things that your body will absolutely want to get rid of and will not want to reabsorb. As we're going through this process in our PCT, 75 to 85% of reabsorption occurs in the PCT. So there is going to be some in your DCT. Not a lot. There is some reabsorption in your DCT, but maybe about 15 to 20%. So 75 to 85% of reabsorption. So that process where we're taking items back into the blood will occur usually through these guys. So then our third process is going to be called tubular secretion, and this is it. Tubular secretion. So tubular secretion is where substances are going to move from the peritubular capillaries and disposed of as urine. So what we're meaning here is that things are going from the capillaries. There's only one P. To the DCT. So this is going from the blood to the nephron. So we're going back. We are now secreting things from the blood to the nephron. Tubular secretion. Up here, that means that all of these peritubular capillaries right here, this is going to be tubular secretion. So it's really tricky to kind of process that. So real quick, filtration is a process where you are going to be filtering out from the blood. You are filtering from the blood. Reabsorption is a process where you are reabsorbing to the blood. So you're reabsorbing whatever you need to the blood. Tubular secretion is a process where you are secreting out of the blood. So you are essentially secreting whatever you need to out of the blood back into the nephron. So when you're looking at this nephron and the process of urine formation, it's easier to either think of it as into the blood and out of the blood, or into the nephron and out of the nephron, because it goes back and forth through a 3-step process. So when you're looking at exercise 40, the goal for this one is absolutely to be able to do activity 1, identify the structures of the urinary system organ, and you do need to be able to do it male and female, but there's only one structure that differs. You need to be able to do the kidney in activity 2, which the majority of this is what we just talked about at the end, which is the nephron. You need to be able to know how the nephron filters blood. One thing I do want you to look at and know is with this kidney is be able to identify figure 40.7, and I believe this is also a drawing. Make sure you are looking at figure A. This is going to be the microscopic perspective of the glomerulus. So make sure you are understanding how that process works. I will not be really reviewing activity 3, which is the bladder. It's not super exciting, so we're not really going to talk about it. But activity 1 and activity 2. So it's not a heavy chapter as much as it is you need to be able to identify structures and know what's going on. So it's a little bit more memorization, but that is going to be exercise 40.